Hello, friends. Welcome to another session of Biochemistry. In this session, we are going to be doing the recall from May 2022 for biochemistry questions asked in INI-CET. Now, this is a series for the quick recall. In these recalls, we are not going into the details of the question or the topic. We are just looking at the question, the options, and the answers. This series is starting from 2020, going right up till May 2024. All the 10 questions, which papers which have been asked during this time, will be revised quickly with the answers. The topics we listed in the analysis of INI-CET, and in that we said 60 main topics, 12 additional topics, total 72 topics which have been asked in last 10 years. We will have separate videos for each of the topics going into the detail of each of the topic, but not in this video. In this video, we are only looking at the question and the quick recall. Okay, so let's start with this set. Question number one. This question has been repeated a few times. DNA protein interaction is studied by. And your options are DNA footprinting, DNA fingerprinting, ELISA, and northern blotting. So for this question, you have to remember the answer that DNA footprinting is the technique used to study the interaction between DNA and protein. The premise of the study is that where the protein is there, the replication of the DNA will be affected. And when you're doing the PCR-like studies, so that part will not be properly copied. So DNA footprinting to study the DNA protein interaction. Next question, which of the following type of inhibition is depicted in the following Line-Weaver-Burke plot? You can see this image here. The blue one is without the inhibitor. The red one is with the inhibitor. Your options are competitive inhibition, non-competitive inhibition, uncompetitive inhibition, and the suicidal inhibition. So please note, in this case, the 1 upon Vmax for non-inhibited and inhibited graph is the same, meaning the Vmax is also the same. Vmax remains the same in case of competitive inhibition. So the answer to this question is this graph is depicting the competitive inhibition. Next question. In a patient with maple syrup urine disease, all of the following amino acids should be restricted in a diet except methionine, valine, leucine, isoleucine. What you have to remember that when you're talking about the maple syrup urine disease, you're looking at the defect in the branched chain keto acid dehydrogenase, meaning what will accumulate is the branching keto acids and their amino acids. The branching amino acids are the valine, leucine, isoleucine, meaning these three should be restricted in a diet. Your answer becomes methionine. Methionine is the answer here. Does not need to be restricted in patients of maple syrup disease where there is accumulation of the branch chain amino acids. Question. All of the following are true about the Human Genome Project, except took nearly 2 decades. Total genes coming out from this 20,500 in humans. Next generation sequencing was used and took international collaboration. Now we have one complete full video on the Human Genome Project, but here I'll just tell you the answer is when the Human Genome Project was started in 1991, the next generation sequencing was not available. It is a sequence, a follow up of the Human Genome Project. Sometime in 2007-8, we had the next generation sequencing, the Human Genome Project being completed in 2006. First is so next generation sequencing was not a part of the Human Genome Project. We'll talk about more about this in a separate video. We'll highlight all the points, all the labs, the outcomes, the fund, etc. Everything will be discussed in that video talking about the Human Genome Project. In Sanger sequencing, the DD and TP are used because options are DD and TP exhibit higher affinity for DNA polymerase, DD and TP exhibit lower affinity for DNA polymerase. Insertion of DD and TP results in prevention of elongation of chain by DNA polymerase. DD and TP cause chain elongation by DNA polymerase. Now see, when I was student, sometimes we used to follow the dictum. If you don't understand anything in the question, the longest option is the correct option. The logic is, if it was not the correct option, why would the examiner go to so much effort to make such a big, lengthy option? And that rule works here. Actually, the correct answer is the insertion of DDNTP is in prevention of elongation of chain by DNA polymerase, because to this nucleotide, a fresh nucleotide cannot be added. The point where the fresh nucleotide has to be added, the hydroxyl group is missing. The hydroxyl group is used for making the linkage, and when the hydroxyl group is not there, new nucleotide cannot be added. We'll talk about Sanger sequencing also in a separate video. This is still used even with the next generation sequencing with some combination to improve the yield. Question: you are expected to produce a protein of interest. Arrange the following steps in the correct order for the same. Your options are lysing a bacterial cell, incorporating complementary DNA into the bacteria, SDS page, protein elution, and hybridization. Please note all the steps involved in this process of production of protein of interest from a bacterial system is not listed here. Only some of the steps are listed here, which creates a lot of confusion. And you can see various fermentation combinations are given here. So what you have to understand, first and foremost, you have to incorporate the complementary DNA into the bacteria. So this one is out. It starts with number 5, not with number 2. Then you have to allow this complementary DNA to integrate with the genome of the bacteria, means hybridization should happen, which is happening here. You don't even need to look at the rest of the options, but just for the record, after the complementary DNA will hybridize with the host genome, then it will undergo transcription followed by translation. When sufficient amount of protein is created, then you lyse the bacterial cell. Now this protein will come into the solution. So you do the SDS page for separation of the protein. And then you do the protein elution by taking out the section in the SDS page where the protein is recorded. Okay. So the correct answer is option B, where first you incorporate the complementary DNA into bacteria. You allow the DNA to integrate with the genome by hybridization. After that, will be transcription translation, not listed here. Then, once sufficient amount of protein is there, you lyse the bacterial cell, then you separate the protein and you take out the. Okay, misincorporation of uracil into DNA is seen in a deficiency of. Now, see, the correct answer here is the folate, because folate is required for conversion of DUMP to TM. Uracil normally doesn't get incorporated into the DNA, but if the folic acid is not there, then in that case, the uracil might get incorporated in place of thymine, because thymine is no longer there. Okay. So please note, folate deficiency can result in misincorporation of uracil into the DNA. Deamination of methylated cytidine residue results in incorporation of which of the following into the DNA? Your options are uridine, adenosine, guanosine, and thymidine. So let's work it backwards. Methylated cytidine, methylated cytidine. What is methylated cytidine? Don't go there. First, there is deamination. So read it like this: deamination of cytidine. When you do the deamination of cytidine, then you get uracil. And then methylation of uracil will give you thymidine. So the correct answer is thymidine. Okay. So if you look at it in straight manner, then it is difficult to understand, but you have to look at it in parts. Deamination of cytidine, spontaneous deamination can happen sometimes, will result in the formation of uracil. This uracil, when it undergoes methylation, then you will get the thymidine. So answer here is thymidine. Match the following transporters with the molecules that they carry for your convenience. I have already matched them correctly. Means hemopexin is carrying the heme. Haptoglobulin is carrying the freed hemoglobin. Suppose there is the hemolysis and hemoglobin becomes free. In that case, it will be transported bound to the haptoglobulin. P will carry the thyroxine. And PLN is sometimes also known as transthyretin, the transporter of thyretin. And albumin is the transporter of the free fatty acids. So if you get different permutation combination, you should be able to answer this question. Icosanoids are formed from, your options are hyaluronic acid, treated irrigation, 4-fused ring, long chain fatty acid. The correct answer here is the arachidonic acid, which is a precursor for the icosanoids. Most commonly, there can be other C20 fatty acids giving rise to icosanoids, but most commonly it is the arachidonic acid. So these were the questions which were there in the May 2022 INI-CET Biochemistry section. Thank you for joining. Keep watching our videos. We are doing the quick revision for INI-CET in the last 10 papers starting from 2020 May, extending up till the 2024 May. So don't forget to watch all the videos in the series. We are also having a separate series for the topics which have been asked in these exams. So stay tuned. Thank you very much. Bye bye.